Okay, so welcome to those who are new to Malaysian Research Insider session, and to those who have joined us previously, welcome back. I'm Ayappa. I'm doing a PhD in molecular medicine at University Science Malaysia, and I'll be your host for today's Research Insider session. And so let's welcome our scientist today, Shi Mun Yee, a research
Yes.
scientist from Cancer Research Malaysia. And I will allow her to introduce herself further on what she's working on. Over to you, Shi Mun.
Okay. Thank you and BIOS for having me today. So my name is Shi Mun, and I'm a research assistant from Cancer Research Malaysia. So I'll be your 10th speaker in this series of talks. And for today's session, I'll be sharing about how we use CRISPR-Cas9 screens to identify new therapeutic targets for head and neck cancer. So I've actually asked Ayappa and Apple to help me set up some questions. So if you don't mind, just answer the questions that will be appearing on the screen, I assume. Just take about 5. How long does this take? Let's see, do you know any famous people? Okay, then everyone knows. Most of the people know a person that has had head and neck cancer. Do you know about? Oh, great.
Okay.
It's nice to see that most of them know.
Yeah.
Most of them know of our organization. So yeah, that's cool. Okay, for the 33% of people who know who's the person who has head and neck cancer, kudos. So for the 67%, I'll let you know in my next slides. So yeah, without further ado, I can just continue, right? So without further ado, let's start today's talk. How do I? Sorry, it's not working. Oops. Okay. Yeah, sorry for the technical error just now. It's my first time using Zoom. Basically, okay, let's start the talk. So before I start my session today, I'd just like to give an overview of how the talk's gonna be like today. So at the start, I'll just give some brief self-introduction about myself and how I am where I am today. And then in the next slides, I will introduce about our organization, Cancer Research Malaysia, and what we do. And then after that, I'll give some background on head and neck cancer before moving into the main topic of today, which is our research project. And before I end my slides, I'll wrap up with some photos that illustrate how it is being a research assistant in CRMY. So so let's see, okay, sorry. So some brief self-introduction. I did my high school in SMK Seri Garing Rawang. And then moving on, I actually did my A-levels in Methodist College Kuala Lumpur. So at the time when I was choosing what degree to pursue, I was very interested in science, and particularly I want to know how diseases happen, like what's the biology behind diseases. So that's why thankfully I got like a JBA scholarship to pursue my degree in biomedical sciences, and I did that in University College London. So for my final year project, I did a project I specialized in cancer biology, and for my final year project I worked on zebrafish models. So before I wanted to further my studies, I felt that I didn't have enough of the necessary lab skills like tissue culturing and also running different molecular assays, because my final year project is just like doing a lot of confocal imaging. So when I graduated, I was very happy to have found an internship at Cancer Research Malaysia that provided the opportunity to learn all the skills that I wanted to know. And then not long after, I was very grateful that I got offered a position as a research assistant for the CRISPR team in the head and neck cancer research group. So moving on, I'll tell you about what is our organization and what we do. So basically, Cancer Research Malaysia is a non-profit research organization in Malaysia, and we're dedicated to saving lives through impactful research focusing on Malaysian population and communities across Asia. So our priority is we want to ensure that Asians are not left out in the fight against cancer. So saying this is because we know that most cancer research is based in the West, and they're done in Caucasians. So basically, we know that cancers that are common in Asian populations would be different from the Westerns. That's why, so we want to focus our research into understanding cancers in an Asian context so that we can develop better therapeutics to benefit our local population. So all of our research work is funded entirely by charities and grants. And then moving on. So the cancers that we focus on includes breast cancer, ovarian cancer, and also head and neck cancer. So, but for today's session, I'll be mostly focusing on head and neck cancers, which is a critical disease in Asian populations. But if you want to know more about what we do in the other cancers, please feel free to browse our website, and I've included the link below. So moving on. It's undoubted that cancer research has helped improve cancer survivorship exponentially. So from 1970, we see like 1 in 4 patients survive for 10 years. And the trend we see is like in 2050, we want to achieve 3 in 4 survivals, patients who can survive for 10 years. But the fact is that this trend doesn't apply to all cancers, and especially for cancers that are neglected or they don't get a lot of research done. So one example is actually head and neck cancers. So what is actually head and neck cancer? It's a group of cancers that arise in the head and neck region. So some of the typical sites include, includes the nasal cavity, the tongue, salivary glands, the oral cavity, and so on, which is indicated in this image here. So what are the risk factors associated with this type of cancer? So this includes like tobacco smoking, betel quid chewing. So in case for locals or the viewers who don't know what is betel quid, so in Malaysia, we actually call it pinang. It's the one that locals like to chew on, and then it stains their teeth red. So yeah, that's just one fun fact. And then the other risk factors include alcohol consumption, as well as viral infections, such as HPV and also EBV viruses. So even though like head and neck cancer is actually the 6th most common cancer worldwide, but yet people are not very aware of what it is, or they don't know about it. That's because it's relatively rare in the West. And like I mentioned previously, it's a very Asian-centric disease. So I'll illustrate my point by showing you. So this is like a world map of showing the different distributions well, it illustrates the number of head and neck cancer incidences globally. So if you look at the colors, the darker the color, it means that there's higher numbers. So it's very prominent that you see that head and neck cancers are quite common in Asian regions, particularly South and Southeast Asia. So that's Malaysia there. And no surprise, majority of the cancer deaths also occur in this region. So if we look into Malaysia, so this is a graph that I prepared showing what are the top 5 cancers in our country. You can see that head and neck cancer is the 4th most common cancer. And addressing the question that I asked just now, do you know any notable figures that was diagnosed with head and neck cancer? So the answer is actually Dato' Lee Chong Wei. So in 2018, he was diagnosed with nose cancer specifically, but luckily for his case, he had the disease, he detected the disease at an early stage, and that's why he could seek early and fast treatment. And I say for his case, it's very lucky, because for most of the patients, head and neck cancer patients in Malaysia, it's diagnosed at late stage. So 70% of them only detect the disease at stage 3 and 4. And this is very concerning, because it's where medical intervention is very challenging, and it's just really difficult to treat the disease. So because of that, the 5-year overall survival is only 40 to 50% for this cancer. So another contributing factor to the poor overall survival is that head and neck cancers really have very limited treatment options. So current standard of care would include surgery, radiotherapy, chemotherapy. So chemo and radio, as I believe most of the viewers would know, it would cause high toxic side effects because it not only kills your cancer cells, it will also kill your healthy cells. So even for what we want to achieve, which is what we want to use for treatment, which is targeted therapy, there's only like 3 options for head and neck cancer, and they also don't show the most effective effects. So I've told you that head and neck cancer really is a major problem in Malaysia as well as Asian communities. And I've just shown you that there is really limited options for this disease. So that's why we think it is very crucial that we look for new targeted therapies to improve patient outcomes in our Asian region. So moving on, so knowing that this has led to why we are doing our research. So what we want to do is we want to look for the Achilles heel of cancer. So what is the weak point in head and neck cancers that we can exploit for treatment? So I've prepared this analogy. So this is a Jenga block. So I'm using this as an analogy for head and neck cancer. So what our team is doing is that we want to look for the essential genes depicted by the blocks here, which are important for head and neck cancer growth and survival. So once we identify these candidate genes, what we can do then is use them as therapeutic targets so that we can topple the tumor structure and potentially cure cancer. So after saying that, just to share how our project approach is. So the first step is that we use CRISPR-Cas9 to identify what are the essential genes that we can target. So after knowing which genes that we can target, then we can look into, we can try and find the drugs that will target these genes. But then knowing just what gene to target and also what drug to use is not sufficient. Also very important that we identify features like genetic features or characteristics that will help us distinguish patient groups who will benefit from this treatment. So point number 3 is moving more into precision medicine. But before I move in and explain how CRISPR-Cas9 screens are done, I just want to give a brief explanation of what CRISPR-Cas9 is and how it works. So it's a very powerful gene editing tool. It's made up of 2 components. So one is the guide sequence, which is highlighted in yellow here. What it does is it helps to locate the gene that you want to edit. And the next important component is the Cas9. So Cas9 is like the enzyme, and it's like the scissors. It acts as the scissors that cut out the gene target. So the concept of CRISPR-Cas9 is basically both of the components work together to find and delete whatever genes that you want to knock out of the DNA. So this is like an animation that my team provided. So essentially, this is a very powerful tool because so with Cas9, it will cut out the gene of interest. So with the gene knockout, cancer researchers can then use this as a powerful tool to identify what are the important genes in tumor cells. So the principle is they cut out each single gene in the DNA, and then they see upon the loss of which gene will this cause the tumor cells to die. So if you cut out a certain gene and that causes the tumor cell to die, this is an indication that that gene is very important. So knowing that, many researchers have been using CRISPR-Cas9 screens to find out what are the essential genes in cancer. So I'm just sharing some of the handful of papers. Like this is just a very, very few of the many that has been published. So CRISPR-Cas9 screens has been done in colorectal cancers, leukemia, rhabdoid tumors, and even pancreatic cells. So, and then these two are actually two big projects which are characterizing the essential genes in many, many different cancers. One of them is DATMAP, and they're from the Broad Institute. And another one is Project SCORE, and they're from the Wellcome Sanger Institute. So having this technology to characterize the essential genes in cancer, we also applied the same technology into our project, but they did a lot on, again, they did a lot of screening in Caucasian cell lines. So what makes us different is that we do the CRISPR-Cas9 screens across a panel of Asian head and neck cancer cell lines, so that we can know what are the important genes that are driving the disease in our local and also Asian communities. So moving on, I'll go through what is the methodology that we do this genome-wide CRISPR-Cas9 screening. So the first step is that, oh, and one small disclaimer, this is an image under publication review, so please be mindful and do not use this image elsewhere. So moving back to the top, the first step that we do is we generate cancer cell lines that express the Cas9 enzyme. So like I mentioned just now, these are the scissors that do the gene cutting exercise. Then after we have these cell lines, we will then infect them with a pool of the guide sequences. So there's about 90,000 guides, and these are targeting 18,000 genes in the cancer genome. So what we want to know is, so these sequences, they are barcoded. So we infect them into 60 million cells and allow the cells to grow for about 18 days. So to explain, if the guide targets a very important gene, it will then, with that gene cut out, it will cause the cells to die. So at the end of the screening exercise, when we extract the DNA and compare it to the initial pool of sequences, we use next generation sequencing to identify which of the sequences that have gone missing from the pool. So if we have this list of guides that are missing, it's an indication that those guides are targeting important genes. And with that, we will be able to catalog what's the important genes that are, what's the genes that are essential for that cancer. So after showing, so currently from gene to drug, we have currently identified a list of candidate genes that we think are important for head and neck cancer and we can use for therapeutic purposes. So currently the next step is to look for the drugs that we can use to inhibit these gene targets. But the journey from gene to drug is actually still a long way to go, because once we've identified a drug gene pair, we will need to validate them in vitro and also have to go through some preclinical and also clinical trials to validate that it's effective to target this gene with its corresponding drug. Then after that, we will be able to hopefully develop a new treatment for head and neck cancer patients. So with that, that's my research project, and these are just some acknowledgements to the institutes and also research grants that enables us to do this research. So like I promised, I will wrap up with some images to show you how it's like working in Cancer Research Malaysia, and also what I do in the CRISPR team. So for the CRISPR team, we do a lot of cell culture work. So this is just an image of, this is the biosafety hood, and these are the incubators we use to house the cells. So you can see like in each flask, this is where we grow the cells. And I'm also prepared some images of cancer cell lines. Yeah, so like doing cell culture work is being like a babysitter. Like you really have to understand the needs of all the different cancer cell lines. And then just to give you a glimpse, this is the image of this is how it was like when we did the CRISPR-Cas9 screening. So yeah, it was like walls and walls of flasks. And to give you a glimpse of how big this the scale was. So this is like, I think that's my colleague there, and you can see that it's on level with her height. So yeah, that was a lot of cell culturing work. And then, yeah, just before I end, this is just some images of how it's like working in Cancer Research Malaysia. So everyone is really helpful. This one, yeah, we had just had this photo recently taken. So we all forgot that we were wearing masks and we just smiled. And we also, like, besides doing a lot of, besides working hard in research, we also do a lot of outreach and activities. So this is, I think it was a campaign to raise awareness for cancer and also for Cancer Research Malaysia. This is a picture of my team. So that's my team lead and also my other colleague. And also we also participate in walkathons and marathons. So that's just us, yeah, being happy with the Milo from the Milo truck. So yeah, that's all. Yeah, thank you.
Yeah, thanks Shi Mun for your very insightful. Okay lah, it's very entertaining. I like the pictures at the back, and I saw the flask. Oh wow, yes.
Yeah.
That's a lot of work. That's a lot of work. Yeah. Okay. So we have few questions here. So let's start with the one on Zoom group chat. Okay. So there's a question here from Apple. So she's asking, how do you choose the 18,000 genes to target?
How do I choose?
Yeah.
I am not, for this, I think for us, we use like established. So these are coding genes, like they code for the proteins. Because I think like theoretically there's about 18,000 genes that they will be made into proteins. And like for our library, we use like the sequences from an established pool already. So someone has already developed that pool.
Okay.
Mm-hmm.
And then we also have we have a general question here. Is like, why are Asians at a higher risk of neck and head cancers? I'm actually very curious about this as well.
Yeah, so this one there's multiple factors. So the one that I know the most is there's some genetics, like Asian and Caucasian genetics are different, but people are still understanding what are the differences that predispose Asians to a higher risk. Another more prominent factor is actually our dietary habits. So for Asians, we tend to take more salted and cured meat. And then we chew, like the locals here, they chew the pinang, the betel quid. So because of that higher exposure, I mean, the Westerns, they don't do as much as us, right?
Oh,
so that's why we get more exposure to the those risk factors.
Oh, okay. Yeah, I was thinking also it could be
habit.
Yeah, it's like, I think it's a habit, and then I also was thinking maybe it's due to our eating habits. Right? Yeah.
And another thing is the EBV infection. But the relationship to how EBV infection causes nasopharyngeal is still under heavy research, because sometimes you can't just say you got EBV and you will be destined to have nasopharyngeal, because it's a common virus that people harbor in the body.
Okay, right then. Next question is, besides nasopharyngeal cancer, what are the other common types of neck and head cancers globally and in Malaysia?
In Malaysia, I did when I prepared the statistics, the next common ones are actually tongue and oral. So actually
Okay.
for our research team, we do a lot of oral research, like oral cancer research as well. Globally, I think in India, they are also, for them, it's like oral cancer again, like oral cavity cancer. But in the West, it's mostly oropharyngeal. So it's like different sites. And then the oropharyngeal is more associated with HPV infection. So that's seen more in the West.
Okay.
Yeah.
Okay. Is it could be, I mean, the next one you said was oral or tongue, right? So it's like, is it because we like to consume hot food? Like always we have to have hot food.
that I don't know. Yes, I've heard that before, but I'm not too sure like whether it's because of that.
Yeah, I mean because if
I'm not sure. Probably because when we take hot food, there's more like we subject them to more damage. But I'm not sure.
Okay. All right. Next question is, so at what stage is this work in? Preclinical trials?
No, it's still in preclinical.
Preclinical.
We've just identified the genes. So now we're working mostly on looking for the drugs and validation in the lab. So we have a few candidate genes in mind, and we have some drugs. I mean, we are still looking for the drugs, but yeah, far from clinical.
Okay.
For now.
Okay, then we have a question here from AE. She's asking, is it possible to repurpose the existing licensed drugs to target your genes of interest?
So
Good
that's a very good question. So that's bang on the strategy. So that's actually one of the strategies that we are, like one of the approaches that we are doing to look for new drug targets, because, well, drug development is very long, painstakingly long and expensive. So yes, we are one of the strategies we are doing that. Like that's why I showed like the drug databases.
Right. Okay, and then another question, we have a question from Duncan, and he's asking, does the cancer cell line translate well to cancer cells found in patients? That's a good question.
That's a very good question, and also a big discussion in our community.
Yes.
Because like cancer cell lines are actually in dishes, whereas you have different environments for like the actual organism. I mean, yes, there is its limitations. It does not I can say that it won't directly mimic our system, the cells in our system, but it's still a very important model that we need to use, because it's somewhere to start. Like at least we have the cancer cell models to work on, and then that's why we do a lot of we have to go through a lot of validations in the lab, and we have to pass through preclinical and clinical trials.
So to add up on that, so do you think that maybe through your in vitro, right? Maybe you can try it's like 3D assays. It will actually portray more.
Human organoids would be the next option. But for now we are still focusing on cancer cell lines. I think projects like Wellcome is doing the CRISPR-Cas9 screen using tumor organoids. So it'd be interesting to see what they find.
Kai Xiang has a question. So he says amazing talk at first, and he's curious that what's the average cost of genotyping per person in Malaysian ringgit?
Sorry, the cost.
Yeah, what's the average cost of genotyping per person in Malaysian ringgit?
Oh, that's a good question. I'm sorry, Kai Xiang, I wouldn't be too aware. Yeah, I will find that answer and answer you on the chat.
I think that's like more to personalized medicine already, and it's going to be like costly, I guess.
I think it's getting, like, we know that genome sequencing is getting cheaper, but yes, it is a costly option.
Yeah. We have a question here from Facebook, Rusley from Facebook. So is there any current clinical trial or application using CRISPR, or is it considered unethical because it involves gene editing?
So the first question, is there any CRISPR-Cas9 treatment? I think there is. If I'm not wrong, it was retinoblastoma, but please don't quote me on that, because I know there is treatments, like they are trying to develop like using CRISPR-Cas9 to treat like as gene therapy. And about the unethical issue part, that's a gray line. I think the one big issue was like recently there was a CRISPR editing baby issue in, I think in
Yes,
China.
yes.
Yeah. So yes, that was an example of unethical applications, because
So
yeah,
trying
it
to
was.
make super babies.
No. Actually, he was trying to prove a point. If I remember correctly, they were trying to. So in AIDS, HIV/AIDS,
Yeah.
there's a very important receptor that helps the virus dock into the cell. So the guy wanted to. He said he could, by removing that receptor, you can actually protect the babies from HIV/AIDS. And it's unethical because I think the parents were contracted the disease, so that's why they yeah, so that's the unethical part. But yeah, it's going through a lot of trials, like trials in court, like not clinical trials. Yeah.
Okay, we have another question. Here is Tom. So is the cancer type classified just because of the location of the tumor, nose or mouth, etc.?
Yeah, because like that's the term that describes the head and neck, the regions in the head and neck.
Okay. Yeah, so I have one technical question here. I noticed that just now you said that the cells were left like for 18 days. Like, why is it
Yes.
like so specific 18 days?
Oh no. Why is it specifically the 18 days? I think it's the time where it's a balance where it's more like, let's see, how do I phrase this? We chose 18 days because you don't want to subject the cells to too much Cas9. Like you don't want it to die because it just kept on cutting. Like you want to have a balance that it's when you cut out the genes and it's lost its function. Yeah, you don't want the cells to die because of excessive cutting. I think that's a straightforward question. I'm sorry if I can't explain this more in a more straight.
No, it's okay. I think it was too technical of me to ask you that question.
No, I should know this answer.
But I did like to add on to that. It's like since it's 18 days, so do you change the media from time? And also because the confluency of the cells is going to increase over time. So 18 days, like what happens in between?
In between, I mean, once we infect the cells, we first have to check that whether if all the cells have the infected with the SG. And then we have to check that. So we check that using the flow cytometry. So once we check that, we have to then, passage the cells. You have to meet a threshold where like we get, you have to grow them. And then if they grow confluent, then we have to passage.
Yeah.
Until day 18, and then we extract the DNA.
Okay. Right. Yeah, let's move on to the next question. So Sean,
okay.
What proportion of NPC patients do not have any identifiable risk factors?
What proportion?
Yeah. Of nasopharyngeal.
This is a very This is a very tough question.
It's okay, take your time.
What proportion of NPC patients have no identifiable risk factors?
Yeah.
I do not know the clear answer to that, but what I know is in Asia, it's mostly, I mean, specifically in Hong Kong, they are more, or like Asian countries, it's more associated with EBV risk, EBV infection. So they use EBV infection as a marker to screen to whether you would have as a potential risk. But yes, I'm sorry, Sean, I don't know the direct question to what's the proportion like.
It's okay, it's okay. All right, it's okay. We just move on to the next question. We have yeah. So she's asking, how long does it take to run a sample using NGS?
How long?
Yeah.
Should be quite. We send our sequences NGS. I think it will be quite
So what,
fast.
1 week, 2 week? Is it 1 week? I think it's okay. So right.
I'm not too sure to how long, because we send our sequences to our collaborator overseas to do the sequencing. I think it's about, we get the results back in a month or so. But yeah, specifically how long, I'm not too sure.
If you pay, you send to company also be faster also, right? If you pay and send to companies, it will be faster. Could it be? I'm not sure. I'm not very sure.
I think a few weeks lah.
Yeah.
It largely depends on processing.
Right. The next question is Cancer Research Malaysia planning to do any genome-wide association study to find genes associated with various cancers?
Yes, I mean, we're actually doing that for the breast cancer research team is actually doing that GWAS. Like because they want to map out what are the mutations that are more associated to Asian breast cancer. And yeah, they are doing it.
Wei Yang has two questions here. Sorry. If the cells which didn't die after the CRISPR or Cas9 editing, does this mean the gene is not responsible for the oncogenic growth?
That's what we think, because like within the 18 days, if you cut out, because for our screen, if you cut out that gene and that cell doesn't die, it suggests that it's not a direct answer, but I think it's suggestive that it doesn't play as big of a role compared to the ones that really depleted, like the ones that once you cut it out and the cells die. Yeah. I hope that answers your question.
Yeah, so yes, he has another question also. How do you obtain the information to select thousands of guide RNA for gene editing?
How do you obtain?
How do you obtain the information to select? How do you obtain the information to select thousands of guide RNA for gene editing?
How do you obtain the information for guide sequences? Yep. Also, do you mean like how do I design the sequences? How do I design the sequences? Okay. For my experience, when I want to design a sequence, there's actually a portal like on Broad that you can use. So it's like a bioinformatic website that you just key in what genes you want to use, and then they will come up with a design, like the optimum design of what guides you should use, and then they rank accordingly.
Okay. Okay, and we have another question from Facebook, Michelle from Facebook. How do you generate Asian cancer cell lines?
Okay, that's a very. How do you derive cancer cell lines from Asian patients?
Yeah.
So thank you for that question, Michelle. So what my colleagues did, how did we develop is we resect tumor samples from patients. Then we process them and we let them grow in the dish. You process and then you let them grow. So if epithelial cells are able to grow and attach to the dish itself, we will continue to culture it until it's stable. So that's how we get the cancer cell lines. But we get but to take note, we developed it using Malaysian patient samples. Yeah. I think there's no difference in how to develop.
Yeah, I think it's all the same.
Yeah, it's all the same, just that we get it from Asian patients and Caucasians get it from Caucasian patients.
Yeah. I think in Malaysia are a bit lucky because we have a very diverse ethnic group.
Yeah, yeah,
So you can
yeah.
get from all different people. Yeah.
Yeah.
Another question from Facebook, Ashwini.
Mm-hmm.
Do Cancer Research Malaysia have
Mm.
collaborations with pharmaceutical companies to test preclinical compounds, and have you been able to move any to clinical trials?
We have collaborations with big pharma. If I am not mistaken, it is Novartis. And then yes, for the breast cancer team, we are starting a clinical trial using Pembro, if I'm not mistaken. So yes.
Another question from Duncan.
Mm.
Wouldn't it be easier to find genes associated with Asian community by comparing RNA sequence experiments between cancer patients found in Western countries versus Asian countries?
Easier to find essential genes by comparing RNA sequences between
Yeah.
the West and the East.
Yeah.
Isn't it easier to get RNA-seq? So you're saying to find the differences between the RNA-seq results?
Yeah.
That's a challenging question, because actually I'm not too how do you say? I don't deal a lot with the bioinformatics side. So I'm more responsible in designing the experiments and more on wet lab. So I don't think I'd be the best person to answer that.
It's okay.
Sorry.
Right, we have another Pei again. Yeah, so she's asking from your viability knockout screen, do any of your significant hits belong to the same functional pathway or groups?
Do any of my hits?
Significant hits, yeah.
Are they found with the known pathways?
Same functional pathways or groups?
Same functional pathways or groups, meaning associated with the ones that we know in cancer, or so it's knockout
associated with the one you know in cancer, yeah. Knockout screen, your viability knockout screen, yeah.
My viability, yes. Actually, from our results, we do know that the CRISPR screen is robust because it is calling out the known essential genes. I mean, the ones that we know that are important for head and neck cancers. But what we want to know also is we want to find potential new ones, so that we can develop those into we can come up with new targeted therapy.
Right. Yeah, I think is that all the questions? Okay, there's one final question. So what's next after Cancer Research Malaysia?
Currently, I'm still focusing more on one option is to further my studies, but I am keeping my options open because I'm also interested in doing like science communication.
Ooh, okay.
Yeah. I like to, yes. But let's see how it goes.
Yeah, only time will tell, right?
Only time will tell, yes.
Oh, wow, we have more questions from Facebook. Are you okay with that?
Okay.
Yeah.
Yeah.
My Sarah from Facebook, she's asking, how do you manage your time in handling a lot of cell culture samples? Is it tedious to maintain them? Nice question.
Oh, that's a good question. Thank you for asking that. Yes, you need to learn how to prioritize, and good advice is before starting the work, it's nice to visualize and plan out how the work's gonna be like, so that when you do it, it will be much smoother rather than just planning it last minute. And you have to be very focused when you're handling tissue culture work to prevent like cross-contamination and stuff.
Yeah, I mean, since you work with a lot of class, Is it often that you get contamination?
A lot of what?
Since you work a lot of people with many flasks at one time, right? So
Oh,
yeah, do you get contamination often?
not too often. Like just abide to the standard procedures. Like minimize processing multiple cell lines at the same time. Do aseptic techniques when you're changing cell lines. So not too much contamination. I mean, occasionally yes, but not too frequent.
Another question is from Shenhan. So, have you found any new gene not previously implicated in head and neck cancers?
Have you found any new gene not implicated in head and neck cancers? We have found candidate genes. Not to say, is it completely novel? A bit confidential.
Okay.
Yeah, it's confidential. Yeah. Sorry.
Bleep, cannot say.
Bleep. Yes. Thank goodness.
Right, now, a question from Jake. Well, two questions actually.
Okay.
Jake, any knowledge of demographics for head and neck cancer in Malaysia? Like more Chinese get it or not? I think yes. Right?
Yeah, it's mostly, I think the top is Chinese and the Indian. Yeah, because of the beetle quid chewing. But I mean, like, sorry?
Chinese people chew beetle quid also, right?
Not Chinese, Indians.
Okay.
Indians is more associated with the beetle quid, I think. But yeah, I think the dietary habits, but it's not just like everyone, not everyone, but it happens across all races.
Yeah.
I guess higher in Chinese and Indians.
The other question by Jake is, which ethnic groups are represented by your cells?
Which ethnic groups?
Yeah.
My cells. I think it's a mix. But that's also confidential.
Okay. Bleep. Sorry.
Yeah, this is patient confidentiality.
Yeah. Okay, next, Ashwini again from Facebook.
Chinese.
How are the job prospects for working in Cancer Research Malaysia, and how would you describe the work culture?
How is the work progress?
Job prospect.
Job prospect in
Cancer
terms of
Research.
No, in terms of opportunities or
I think she's asking for opportunities. Yeah.
Opportunities would depend on the availability and whether we have availability and project availability mostly. I think currently it's good to start for me. Job prospects. I think it's okay. Like you can try, but yeah, like I said, it's subject to project availability. But you can always try for internships.
Yeah, I think that's what they are going for, because now it's like the internship season. So everyone wants to try for internship. So do you think Cancer Research Malaysia have a lot of positions for interns?
Also depends on how's the projects like, and whether there is available allocated funds to hire interns. But I'm not the best person to ask that. That's HR. But yes, just try.
Yeah, her other question was, how do you describe your work culture there?
My work culture, it's really nice to work there. Everyone is very helpful. Very helpful. And it's a nice bunch of people to work with.
Wei Yang, okay, another question by Wei Yang. Upon screening of the knockout cells, what's the next experiment do you do after? Do you study the specific effects of the knockout genes in cancer cells?
Okay, so after we knock out, then what do we do next? So after we actually do the CRISPR-Cas9, we then have to do like bioinformatics and really narrow down which are the genes that, we have to select which are the genes that we want to prioritize. So if we have a gene candidate, the next step is to actually knock that gene out, like just knocking one, like in a single manner to show that it really is important. So after that, after knocking that, then we do like from literature review, we will then do multiple essays, like depending on what question you want to ask. So we'll be doing a lot of qPCRs and Western blots. Yeah, just to validate the importance and how is it, like what mechanism is it involved in? How is it important for the cancer? How is it playing an essential role in head and neck cancer development? Yeah.
A question by Zhen Jie. I think maybe
Okay.
this might be the last question.
All right.
Zhen Jie, has your group looked into non-coding genes, for example, like long non-coding RNA?
For now, no. The ones that we screened are directed to coding genes. I think the non-coding genes are more complex. Yeah, not as straightforward as the coding ones.
Okay.
Oh no, we have not done this yet. We have not done this yet.
Yeah, I mean, it takes time, right? Because I can see that you
Yeah.
have so much work to do with all those cells. Wow, that's crazy.
I mean, the screening has passed. Like now it's more to like looking for the narrowing down to the candidates for validation. Yeah.
Okay, I think that's it for the questions. So
Mm-hmm.
yeah, so thank you very much for sharing your research with us, Shi Mun. And
Oh, I
thank
hope it was
you
helpful.
to everyone who have joined us today. Next Saturday, we'll be joined by Dr. Izzat, who will be speaking on nanomedicine and drug delivery in cancer. And for more updates regarding future Research Insider series, like our Facebook page, MBIOS, and also 100 Scientists of Malaysia. So thank you, everyone. Thank you very much, Shi Mun.
Thank you for
See you
having
next
me.
Saturday. Yeah, thank you. Bye-bye.
Bye.